More peculiar than the concept of friction is the enigmatic force that serves as the catalyst for the emergence of life. The propulsion of development in a marine organism is thoroughly examined by scientists with a focus on the forces of friction. Greetings to everyone. In today's video, we will be discussing a fascinating discovery made by scientists from the Heisenberg Group at the Institute of Science and Technology, Austria. They have uncovered the significant impact of friction forces on the reshaping and reorganizing of sea squirt oocytes following conception. These findings shed light on the crucial role that friction forces play in shaping and molding a developing organism. By examining the influence of mechanical forces on cell and organismal structure, this study offers valuable new insights. So, without any delay, let's dive into our discussion. In the process of molding clay on a spinning wheel, the potter utilizes the friction between their hands and the pliable clay to transform it into a myriad of shapes and designs. Similarly, within the sea squirt oocytes, there exists an intriguing parallel where friction is harnessed within different compartments within their interior to undergo developmental transformations following conception. The Heisenberg Group at the Institute of Science and Technology, Austria, ISTA, has recently conducted a study published in Nature Physics that delves into the intricacies of this phenomenon and elucidates its mechanisms. Underwater, a captivating array of life forms can be found in the sea. From vibrant fish and algae to marine snails and sea squirts, an entirely distinct world awaits exploration. Sea squirts, also known as ascidians, possess a remarkable peculiarity. They undergo a phase of free movement during their larval stage before settling down and attaching themselves to solid surfaces such as rocks or corals. It is during this stage that they develop their defining feature, tubes or siphons. Despite their adult appearance resembling rubbery masses, sea squirts are actually the invertebrate relatives most closely linked to humans. Surprisingly, sea squirts bear a striking resemblance to us, particularly during their larval stages. Ascidians being creatures that possess both the fundamental developmental and morphological characteristics of vertebrates, as well as the cellular and genomic simplicity commonly found in invertebrates, are frequently utilized as model organisms for the examination of the initial embryonic growth of vertebrates, including humans. Carl Philipp Heisenberg, a professor at the Institute of Science and Technology, Austria, ISTA, elaborates on this by stating that the ascidian larva, in particular, serves as an exemplary model for comprehending the early stages of vertebrate development. In a recent publication in Nature Physics, the latest research conducted by his team sheds new light on the development of ascidian oocytes. The findings indicate that friction forces play a vital role in reshaping and reorganizing the internal structure of these female germ cells after fertilization, marking the beginning of their developmental cascade. The process of cytoplasmic reorganization, which occurs in animal oocytes following successful fertilization, involves the alteration of cellular contents and components. This reorganization serves as the foundation for the subsequent development of the embryo. In the case of ascidians, this rearrangement results in the formation of a bell-shaped protrusion called the contraction pole, CP, where crucial materials necessary for the embryo's maturation gather. Despite its significance, the underlying mechanism driving this process has remained a mystery. To unravel the enigma, a team of researchers hailing from ISTA, Université de Paris-Cité, CNRS, King's College London and Sorbonne Université embarked on a mission. To aid in their quest, the Heisenberg Group imported fully grown ascidians from the Roscoff Marine Station in France. Nearly all sea squirts possess both male and female reproductive cells, making them hermaphrodites. Within our laboratory, we house these creatures in saltwater tanks, ensuring they are kept in conditions that mimic their natural habitat. This allows us to extract eggs and sperm for the purpose of studying their early embryonic development, explains Silvia Caballero Manchebo, the lead author of this study and formerly a PhD candidate in the Heisenberg lab. Through microscopic examination of fertilized ascidian oocytes, scientists observed a consistent pattern of cell-shape changes leading to the development of the contraction pole. The initial inquiry by the researchers concentrated on the actomyosin cortex, a dynamic structure located beneath the cell membrane in animal cells. Comprising actin filaments and motor proteins, this cortex typically serves as a catalyst for cellular shape transformations. According to Caballero-Manchego, the movement and shape changes of fertilized cells are attributed to increased tension in the actomyosin cortex, which contracts and causes flow. However, the flow of actomyosin ceases during the expansion of the contraction pole, indicating the involvement of other factors. To investigate further, the researchers examined other cellular components that may contribute to the expansion of the contraction pole. During this exploration, they discovered the myoplasm, a layer consisting of intracellular organelles and molecules located in the lower part of the Ascidian egg cell. Caballero-Mancebo describes this layer as a flexible solid that undergoes shape changes alongside the oocyte during fertilization. As the actomyosin cortex flow takes place, the myoplasm undergoes folding and generates numerous buckles as a result of the friction forces that arise between these two components. Once the movement of actomyosin ceases, the friction forces dissipate as well. According to Caballero-Mancebo, this cessation ultimately causes the contraction pole to expand as the various buckles in the myoplasm resolve and transform into a distinct bell-shaped bump. This study offers a fresh perspective on the influence of mechanical forces on the shape of cells and organisms. It highlights the crucial role of friction in the process of shaping and molding a developing organism. However, our understanding of the precise function of friction in embryonic development is still in its infancy. Heisenberg further emphasizes the intriguing nature of the myoplasm, which is involved in various embryonic processes in ascidians. Unraveling its unique material properties and comprehending their impact on the formation of sea squirts holds great interest. To support our channel's growth and ensure broader awareness, kindly hit the like and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.